Okay, thank you. So, a very good afternoon to all of you. It is my proud privilege to introduce before you the speaker of today's presentation, Dr. Abhishek, who is working as a senior scientist in the division of Bacteriology and Mycology, ICR IVRI, Izzatnagar. And he is an experienced veterinary microbiologist working particularly in the area of a lesser investigated field, that is the mycology. Now, the briefly about his biodata, he has graduated from GB Pant University of Agriculture and Technology, Pantnagar, and he has done his master's and doctorate degrees from IVRI, Izzatnagar. And currently, he is looking after the mycology lab, leptospira lab, and the HS lab of the division. And he has handled 14 projects as PI or co-PI of various funding agencies, and currently he is handling 7 projects as PI or co-PI. And his area of research is animal and zoonotic mycosis and bacteriophage research, especially on its therapeutic and vaccine potential against Salmonella and E. coli. He has published more than 40 research papers in national and international journals, and he has 35 other research publications to his credit. He has guided 4 masters and 1 PhD student, and currently 4 PhD students are under his guidance. His main research focus is on the study of diversity and heterogeneity among animal and human dermatophytes, analysis of phenotypic and genotypic antifungal resistance, and virulence typing of animal isolates of the dermatophytes. He is a member of the International Working Group of the International Society of Animal and Human Mycologists. And he has won many awards like the Young Scientist Award, the Best Student Award, and he has been invited as subject matter expert to deliver lectures in various national and international conferences. So, with this brief introduction, I invite Dr. Abhishek to present his talk on recent advances in fungal disease diagnosis of animals. Dr. Abhishek, please.
Thank you, Dr. Rinku ma'am. Uh, Good afternoon to everybody. As introduction has already been given by Dr. Rinku, that I am working on the field of mycology, which is highly neglected area. Still, we are trying to make it important. And so today, the topic what I am going to deal with is the recent advances in the fungal disease diagnosis of the animal. So I guess everybody knows this, that the most of the fungal infection in animals are very common, but they are undervalued and underappreciated. There are a lot of environmental changes are happening that leads to the emergence and widespread of the many other fungi which were not earlier reported. And the most important thing is that why these fungus are important, because they are normally transmitted from animal to human or vice versa. Normally, the outcome of the interaction between the fungus and the host depends upon the health status as well as the age of the host. And these are the two key factors in the progression of the disease or the outcome of the disease. As you all know that the immunocompetent animals or immunocompetent humans, they may not suffer from the fungal diseases, and these fungal diseases only occur when there is a compromised immune system. So mainly the fungal infection can be divided into opportunistic and acquired. So most of the time, these opportunistic infections, they're not causing any diseases. Those infections which is acquired either from the animal or human, they are the real threats. So the main diseases caused by these fungus are the fungal mastitis, abortion, mycotoxicosis, and there are a lot of many other diseases which you might have heard about. So these are the important fungal infection. We are not going into the detail, but these are the most important fungal infection recently or has been in past has been reported from the various animal, including the large as well as the pet animal, like dermatophytosis, aspergillosis, mucormycosis, candidiasis, cryptococcosis, histoplasmosis, rhinosporosis, and the most important is the mycotoxicosis, which is caused by the mycotoxins. So I am not going into detail, just we'll have a brief introduction about the every important disease like dermatophytosis, which is very important in animal as well as the human. And nowadays, because in the last 10 years, there is a change in the clinical outcome of this dermatophytosis, or which is probably known as a ringworm in the humans. And they are in the thought of that maybe there is a possibility that this change in the clinical symptom or clinical lesion or clinical pattern is because of the transmission of the animal dermatophytes to the human. So these ringworm, as you all know, they affect the superficial layer of the body, and mainly the dog and cats are affected. Though large animal, there are very few reports from the large animal, the reports of dermatophytosis. But still, the large animal is also the main source of the infection to the humans as well as to the other entity. Now, earlier there was the report that we have only three genera: Trichophyton, Microsporum, or Epidermatophyton. But now, as per the recent classification, there are seven genera which I have mentioned here, and they have been classified into the zoophil, anthropophil, and geophil. Everybody knows about these zoophils, anthropophils, and geophil, which are very very important. Sometimes there is a change in the host barrier, and geophils may affect the animals and as well as the human, and anthropophil may also affect the animal. So the mainly clinical manifestation depend upon the host immune status, virulence of the particular fungus, and the various managemental practices what we are following in the farm. So these are the few pictures from our lab where we have seen this type of the infection from our farm where we have seen this type of the infection is the typical ringworm infection at the we can say like the at the muzzle area and at the near the eye orbit. And these are the few microscopic picture of the microcritia of the microsporum by which we are making a diagnosis of these dermatophytosis. Second is aspergillosis, which is localized fatal disseminated disease, mainly affecting the respiratory system, and most important agent is the Aspergillus fumigatus, and mainly it affects the lungs. And most important host affected by this disease is the poultry, and you might have heard about the bloated pneumonia, which is important in the poultry, and sometimes there is a complete stamp out of the flag happens when this aspergillosis used to be spread into the flock. They also affect the ruminant and they also affect the equines. So these are the few pictures where we can see the postmortem lung of the poultry, and here we can see the typical picture of aspergillosis when poultry suffers. They usually do the gasping, and these are the identification microscopic slides for the identification. So now the mucormycosis, it is also a very important infection, fungal infection in the human as well as in the animal. And the most important thing here is that these diseases only spread when there is a complete immunodeficiency or there is a breach in the immune system. So they also affect the almost all animal, and they also involve in the systematic infection. There is a myth that the mucormycosis is or mucus mainly causes superficial infection, but they also are important agent in the systematic infection also. So almost all animals used to get the infection from get infected from this mucormycosis. Now you might have heard about the role of the black fungus in the COVID patient, where the means a lot of havoc has been created that the black fungus is causing like havoc, especially in the COVID-19 or recovered patient or the recovery patient among the humans. So this is the most important among the black fungus, or we can say the these are the mucorales, where the most important fungal agent is the Cunningham mela-Bartholeti, though it was not reported during the recent outbreak of the COVID-19. But this is the most important agent which is causing the highest mortality among the patient, among the COVID patient, or not only the COVID patient, any patient which is suffering from the pneumonia or from the other diseases. So the most important predisposing factor for this disease is the administration of the corticosteroids, like some of the drug like tocilizumab, and there is also reports of hematological malignancy by this mucorales, or we can say like by the Cunningham mela or other mucorales like Mucor rhizopus and Absidia. So candidiasis is also an important disease which is causing the mastitis, is also involved in the abortion. They can also cause the superficial diseases. They can also cause the systematic infection. Most important agent is the Candida albicans, and almost all animals used to get the infection, but mainly the dog and cat, where they are causing the cutaneous candidiasis, and they are also important agent causing the otitis media and otitis externa. They can cause the superficial disease as well as the disseminated disease, where they can be a main agent in the myocarditis, endocarditis, and endophthalmitis. So you might have heard about the Candida auris infection, which is classified as a emerging fungus, and this is the first fungus which is causing a acute death, and it is a serious global health threat declared by the CDC. And the main because of three main reasons: because it is a multi-drug resistant and is very difficult to identify, and there is a possibility that it is misdiagnosed with the other Candida albicans, other Candida agents like Candida albicans or parasimplosis. Now, another disease is the cryptococcosis, which is very, very important in the human as well as in the animal. And you all know that is the pigeon dropping is the main source of the infection of the cryptococcosis, and is also known as the Caverian disease that used to occur in the, like in the Caverian area. So this is also known as the Caverian disease. Mainly the dog mainly the cats are affected by this, but there are few reports of the dog as well as the horse. They are also involved in the mastitis and the respiratory infection. Histoplasmosis is another disease which is very rarely reported in animals, but it is common in the human. And the main source of the infection is the inhalation of the fungal spore, either present in the soil or in the environment. And there are two species which is very important. One is the capsulatum and another dubosi, and they are mainly involved in the reticuloendothelial system in type of the symptoms. Now, rhinosporosis, which is very common in the southern as well as the northern part of the India, caused by the Rhinogenes sebarii. It is a chronic subcutaneous infection characterized by the typical polypoid growth and on the nasal cavity, or maybe the other cavities like in the ear, or maybe in the eyes, or maybe in the vagina even. So everywhere this like any cavity can be affected by this rhinosporosis, and they usually characterized by the polypoid growth. This is very, very endemic in the Tamil Nadu, Andhra Pradesh, Odisha and West Bengal, though few cases have been reported from the northern part of also, and there is also possibility of disseminated infection in the human through the hemateginous route. And another aspect of the fungal disease is the mycotoxin. So mycotoxicosis, there are a lot of mycotoxin which are causing different type of the affecting different type of the organs, like some are hepatotoxic, some are the genotoxic, some are the teratogenic. So we are not going into details. So our main topic is to what are the advances, or what are the method to diagnose these fungal infection. So now I will come on that. So fungal diseases, I have already told that they can be classified into the superficial, subcutaneous, or systematic diseases. Mainly in the animal, people are concerning on the, or we can say focusing on the superficial diseases. Nobody is bothering about the systematic diseases. As this systematic diseases like aspergillosis, histoplasmosis, or many other diseases, they are very difficult to diagnose. They are very difficult to differentiate from the other diseases. And few of the fungal diseases can only diagnose only at the necropsy. So there is a little attention towards that. So the timely identification of this fungal disease is very, very crucial, especially in the systematic diseases, and to initiate the proper antifungal and suitable antifungal therapy, because sometimes this diseases leads to the irreparable damage in the host body. So normally, or we can say conventionally, we are diagnosing this fungal infection by the conventional method, like by we are doing diet microscopy, we are doing culture, and then we are most of the cases we are diagnosing it by the conventional method in the lab. But these methods are now we cannot say they are obsolete, but they are having some limitation, because now everybody wants the result in a quick time. So it is very difficult to give result in a very short time, because these methods are time consuming, because every fungus used to take a lot of time to grow. So there are introduction of few molecular assay and serological assay PCR, genome sequencing, multi analysis, and these assays is not only used for the diagnosis or for the quick diagnosis, they are also used for the understanding the diversity as well as the heterogeneity existing among the isolates. So there are few serological tests where they are detecting the antibodies for that particular agent, but they are not very conclusive, because most of the fungus, systematic fungus, they usually share the common antigen. So there are a lot of advancement has been done, especially in the diagnostic field. And if we will talk about the mycotic disease, there has also been good advancement has been done. But main means, like we can say, the main limitation is that the people, nobody or no clinician is suspecting that it may be a fungal disease. So here, my contention is that if a patient or if animal or a human is referring to you, and if he is not responding to the treatment or to the antibiotics or to the alternate antibiotics, then we should suspect that there may be the possibility of fungal disease rather than to the other diseases. So if we are suspecting there is a fungal disease, then we can at least we can evaluate our diagnosis or prognosis, and we can think of that there may be the possibility of the fungal infection. So there are many advanced method which has been now almost established to diagnose the various fungal diseases, like PCR-based method, where we can use different sepsis test, light cycler, septifast test, film array. Combined, there are combined method where we can combine the yeast and mold together, and we can make it a diagnosis. Like Eplex is the one method where we are diagnosing the mold as well as the yeast in a single test. There are very many conventional tests where we are provided with a chromogenic media, which normally gives a different type of the color, and on the basis of the color or pigmentation, we can identify that particular fungus. We can do the microscopy at what we are most commonly following, and then we also go for the histopathology staining, where we can diagnose with the presence of the hyphae. We can also go for the serological methodologies like fungital assay, lysa, and these are the commercialized assay. And most important thing is that means like they have commercialized, but still their accuracy, sensitivity, and specificity is in the out. So we can also use mass spectrometry like MALDI analysis. We can also do the hybridization technique. We can also use some non-magnetic resonance technique like T2 Candida panel, which is very important for the diagnosis of candidiasis or differentiating the different candida strains. Main thing is that with these all tests, there are few limitations. Some are time consuming, some are not commercially available, some may require the growth of the culture, some may be only applicable to the clinical sample, some need some mycological evidence to move on further. So these all tests together is like, we cannot say that a single test or single any advanced test is enabled to do the exact or accurate diagnosis. There is a We should always approach with a combined diagnosis, conventional as well with the molecular test or some other technology test based test. So rapid and better diagnostic is required, is the need of hour now, because to start an early antifungal therapy, and we should not go for the we should not go for the wrong antifungal therapy, because this and antifungal antibiotics they are very costly and they are very toxic. So means if we are doing this diagnosis at early stage and better diagnosis, then we can avoid this use of unnecessarily or we can avoid this use of not required antifungals, so that the animal should not suffer with any type of the toxicity or maybe the liver in suppression, or we can also reduce the mortality rates among the animals or maybe the humans. So there are few systems, automated system have been developed, like for the bacterial identification. These are the based on the turbidometry, and where we detect the biochemical activity of particular fungus. And the main limitation with this automated system is that they require a lot of data to compare. So about the fungus, if you talk, there is a less data is available. Data is very scanty, and though from human side it is lot of data, especially on the candida, dermatophytes, aspergillosis is available, but for other fungal agent like rhinosporiasis, leptoplasmosis, there is less data available. So these tests we can use it. Mainly they are used for the yeast identification, like API 20C AUX and Vitek system, but we can also use for the few mold like dermatophyte we can use. For aspergillosis also we can use this automated system. Mainly they mainly concentrate on the yeast type of the yeast identification, especially the Candida and maybe the other yeast like Cryptococcus. Then we can go for the nanogymology, where they use the nanozymes, and these are the particles which are having a mimicry with the real biological enzymes, and by tagging with this with the suspect agent, we can diagnose it. Like normally, this is used for the diagnosis of the gelanon fungal toxin, which is produced by the Fusarium, which is very important uh, toxin in especially in the pigs. And then aflatoxin, which is very important in the pig, responsible for the Brooder's pneumonia or other type of the mycotoxicosis, that we can diagnose it with the use of these nanoparticles, or we can say the nanozymes. So there are few differences. It has been quoted that they have used for detection of aflatoxin by using the ELISA or immunosorbent assay, and then by calorimetry also they have detected this gelanon at the early stages. Now, volatile organic compound based assay also, as every fungus used to produce some unique volatile organic compound. So on the basis of that, they used to detect, they used to find out or to differentiate the fungus. Especially this method they are applying, especially for the Aspergillus, where they can differentiate between the Aspergillus fumigatus, Niger, and many other species which are affecting animals or the humans. So they normally Aspergillus produces different isolate produces a different type of the volatile organic compound. So this method can be used for the diagnosis of the aspergillosis also. Now the lamp also, it is very common nowadays. People are using for the diagnosis of the fungal diseases as well as the bacterial diseases, where we are targeting the important 6 to 8 distinct regions in the target DNA. They are making a it is a method for the rapid visual identification. So with the help of the cyber green, we usually detect by our naked eye that this lamp assay is a positive or not. Mainly they are using for the candida infection affecting the tooth, or we can say the mouth. So this the accuracy of this test is not we can say, like, they are not very sensitive, and there is a lot of non-specificity is there. But still, for the few dermatophyte like Mycosum canis, Trichophyton mentophyte, Fumonisin is the toxin, they are using this lamp assay. Now, multi-analysis also we can do. As for the bacteria, here we usually generate the peptide fingerprints, or here, or maybe we can say for fungus also we can do, like bacteria they are using, and here we normally we normally use the mixture of matrix and analyte, and on the basis of the matrix analyte reaction, on the basis of the kinetic energy, we usually detect the here mass to charge ratio. And normally we are this multi-analysis we are using for the bacteria. There are not many reports of its use against the mold, especially the Aspergillus or dermatophyte, but there are few like papers are available which are commonly using this multi-tophan method for the identification of the resistant strain of the Candida auris as well as the albicans. So there is some nanoparticle based assay is also there, where we can tag the gold nanoparticles with our clinical sample, and then we can detect the change in the color. Here you can see in a picture where the gold nanoparticle is along with the clinical sample. It is changing, giving some different color and different pattern. So by this also we can diagnose the fungal infection, but it is again not very sensitive. But still, to strengthen our diagnosis or to supplement our diagnosis, we can go for this nanoparticle based diagnosis also. Now, solid phase cytometry, this is not very commonly used for the diagnosis of fungal diseases, but for the Candida, as this Candida and Cryptococcus, they are yeast, so they normally behave or grow or having a biochemical characteristic like bacteria. So most of the techniques, advanced technique, whatever is applicable on the bacteria, they are applied onto the Candida also. So here we are doing the quantification where individual cell can be detected on the basis of the fluorescence. So normally they are using for the Candida albicans, where we can examine this cell labeled green with the help of the solid phase cytometer. Now Doppler digital PCR, where we are amplifying each droplet of the sample, and we can use it for the mold as well as the candida or maybe the yeast. Normally, this Doppler digital PCR have been used. Not much reports are available, but they are using for the diagnosis or differential diagnosis of the Aspergillus teres from the respiratory sample and Aspergillus from the environmental sample. So where we can detect the Aspergillus species affecting the animal, or it is present in the soil. Then we can also detect the candidemia with the help of this Doppler digital PCR, as we are using here the droplet which is going to convert. So this Doppler digital PCR is not very commonly followed, but we can use it. So these are the few. There are a lot of many other methods I have mentioned, because it is a very difficult to describe or to present here all method. I have mentioned all in the write up, but still there are many challenges with this advanced techniques. So means the combination of the advanced and molecular or conventional technique is the best way or best approach to diagnose the fungal infection. So there are challenges like few are the pathogen which are changing, which are emerging, or which are having their different type of the antigenic structure, or there is a possibility of generation of the new strains. So where these methods are not applicable, pathogenesis and immune mechanism, the interaction of the fungus with the immune system or with the host is still not very clearly understood. For many of the fungal diseases, there is a cost of factor is always applicable in the veterinary side. So there is a very like these are very costly techniques. They need a lot of money, lot of expertise, lot of human. Technically, they need some like expertise, uh, technical officer, and so the diagnostic in the field of antimicrobiology is still in the infant stage. So we are relying normally on the conventional technique, and though nowadays we are combining our conventional diagnosis with the few advanced technique which we have done in our project also. So this is the means like brief introduction about the mycology, which is highly I guess nobody is working or nobody is having a good experience with the work on the mycotic diseases. So this is a nutshell about the mycotic diseases, and how we can approach to diagnose it, so that we can initiate an early or suitable antifungal therapy. So if I am allowed, I can present few slides what work we are doing and what work have been done in the past.
Yes, yes, sure, Dr. Abhishek,
Yeah,
please.
thank you. So in the IVRI, we are running few projects on the mycology. This is the project which is ICMR funded, where we have targeted the Trichophyton mentagrophyte and Microsporum canis. And in this project, we were having an objective to find out if there is any linking of the human and animal isolates. So these were the two objective, and we are having a collaboration with the PGMI in Chandigarh, where Dr. Shiva Prakash is a very renowned mycologist in the India, was the co-PI. And this project is summarized in the month of September. So in this another project, what we are doing, we are also evaluating the cow urine on the of its and its antimycotic effect. Another project on the dermatophytes is we are also trying the samithosome as a novel transdermal delivery system to check whether they are having any antidermative effect or not. Now this is the one project which we have completed last 2 years before, where we have collected the sample from all over the India, and we have characterized all isolated agents biochemically, and we have studied their heterogeneity as well as their antifungal resistance. Now these are the few pictures where the dogs, where the animal, or we can say the calf, dog, buffalo, they are having a typical lesion of the dermatophytosis, or maybe the other skin diseases. Like these are the few pictures we have collected during our project, and you can appreciate the clear cut lesion. And normally it was earlier myth that this dermatophytosis or the other fungal disease affecting the skin, they usually localized, or they usually cause or produce the lesion at particular area. Now the skin diseases, especially the dermatophytosis, they can cause infection at any area. They can even cause the systematic infection also. There are few reports that a human which are earlier suffering from the localized infection of the ringworm, they got the generalized infection, they have been recommended for the complete bed rest by the doctors. So this dermatophytosis or ringworm is nowadays is a havoc, especially among the humans. So here we have done almost all biochemical characterization, and then we have done analysis, phylogenetic analysis also of malassezia, which is another important yeast affecting the skin, and where we found that the almost all the means like isolates of animal are having a distinct cluster from the human. So like Malassezia pachydermatitis, which is also common in the, or maybe found in the human, but is mostly affecting the animal, especially the canine and the cats. We've also done this phylogenetic analysis for the Candida albicans, where most of the animal isolates are as having a similarity with the isolates of the human. And we have also generated this heat map for the antifungal susceptibility test pattern, where we found that almost Aspergillus, especially from the poultry isolates, they are resistant to the antifungal drug. Though Candida isolates are pure sensitive, but still we have found that almost all isolates were showing the multi-resistant. We have done this analysis for the dermatophyte also, where we found less resistant, but still out of like around 25% of the stains were the resistant. So by this, what we are doing in our lab is usually we are following the conventional as well as the molecular method for the diagnosis. So once we go for the conventional diagnosis by the microscopic test, by the hair perforation test, by the germ tube test, for the candida, many other tests are available. Those are classified under the conventional test, and then we followed the for the confirmation, maybe the PCR or multiplex PCR or RT-PCR, and followed by the if required for the sequencing. So these are the few typical pictures from our lab, where like we are working on almost all fungus, but concentrating on the dermatophytes. So we have done all characterization, molecular characterization. We have standardized all the PCR by our self-designed primers for different gene, like for the pandem, then tripophyton specific. Then we have targeted the TEF1 alpha, beta tubulin, and microsporum specific also. PCR we have standardized. These are the PCR pictures. We have also done the Virlo typing, and very interesting finding is that most of the resistant strain is having a good expression of the virulence gene. Like fungalicin and subtalicin are the two important genes in the dermatophyte as well as in the other fungi. And this is the very interesting finding that the resistant strain is having a more virulence also. So we have also done the this sequencing by targeting ITS and beta tubulin and TEF1 alpha gene. We have also done the whole genome sequencing of the few isolates, and we have found that there are several we can say the sequence types are visible. We have also done standardized this AFLP technique in our lab for the differentiating the trichophyton mentogroup phyte isolates, and we found that the most of the trichophyton mentogroup phyte isolates, they usually belongs to the same cluster where the human isolates are falling. We have also done the antifungal MIC, antifungal susceptibility testing, where we have calculated the MIC, and we found that that most resistant drug is the fluconazole, which is very commonly used in the human for the ringworm. So here we found that almost all isolates were showing the resistance to the fluconazole. And now I will recommend that when if you are anybody is experienced with the experiencing or having a ringworm infection, please do not take a fluconazole, because we have so few human isolate also, and we have tested the fluconazole is having a very high MIC. Then these are the few papers what we have published from our few projects. One in the medical mycology, one in the Brazilian Journal of Microbiology, one in the Journal of Fungi, which is high impact journals. And these are a few other publications what we have done from our work till now. And these are the few unique sequences we have submitted in the NCBI, as well as in the VTCC, and as well as in the CBS HM database, where few of the sequences, like from Malayssia pachydermatitis, we have submitted first time from the India of the canine origin. So NCBI has given us a certificate also that these are the sequence first time submitted from the India of canine origin. These are the few things which we have done for our students. And so this is the nutshell, and we are working on mycology, which is highly neglected area. And these are the few future perspective which we should always take into the consideration whenever we are trying to work on the mycology, or we are trying to work on the micro disease of the animals. Thank you very much.
Thank you very much, Dr. Abhishek, for your very informative lecture and excellent presentation on mycotic diseases and their diagnosis. And I'm hopeful that at least some of the participants they might develop interest and they'll get motivated to study mycology because it has significant zoonotic potential as well. And you covered all the areas, and the very good part was that you also showed the overview of the research activities being taken in your lab. So I'm hopeful that the participants they might have benefited. Now I request the participants to please ask any question from the speaker.
Thank
They
you,
could,
ma'am, for your nice word. Thank you very much.
it was really very good lecture.
Thank you.
The participants, if they can write in the chat box or they can directly ask Dr. Abhishek. I request the participants to ask few questions because it is indeed a neglected area and we do not take it seriously to study. So I think there will be some questions from some of the participants at least.
That's what when they do not have any question.
Yeah.
Yeah, my one thing I want to say that though this disease like mycotic disease are not very important, or we can say like they are not causing any direct mortality, or they are not affecting him much, but they are causing indirectly very like economic losses to the livestock as well as to the
Yeah.
pet.
Yes, yes.
If you are diagnosing this skin infection at the time, or if you are suspecting that your animal is suffering from the skin infection which is not responding to the antibiotics, immediately should suspect that there may be the involvement of the some fungus or some mycotic agent, and you proceed in that direction. And I will request, like, you can ask your clinician also that he should also suspect the that there is a possibility of the mycotic disease. So that is a one way, because earlier in IVRI, we hardly getting 2 or 3 sample for the suspicion of mycotic diseases. When I started work, I used to visit the clinics and to other other animal hospital also, where I used to collect the sample, and I found that there is a lot of positivity. Occurrence is like very good. We can work on it. But
Yes, definitely. Yes.
yes, ma'am.
Yes. There's one question. Which fungus they are more infectious for humans?
Ma'am, actually, fungus almost all is a zoonotic. All can be transmitted from the human, from animal to human, and vice versa. But I will tell you about the dermatophyte. Earlier, it was reported that the trichophyton mentagrophyte, which is a very common agent in the human ringworm, is also a main agent in the animals. But this is not a picture. With the ICR project, we have concluded that trichophyton mentagrophyte, though it is a present in the canines, but it is not a main agent. Main agent is the Microsporum canis, and especially the subspecies of geophilic species like Microsporum gypsum. When Microsporum gypsum causes the infection in the animal or in the human, they are very dangerous. They usually gives the typical lesion. Typical lesion means they like the most of the human, most of the animal, they do not experience the typical lesion of the ringworm because they will start some antifungal or some antibiotic along with the antifungal, so that lesion used to be suppressed.
Yes.
But this, when Microsporum gypsum is a crossing its host barrier and causing the infection in the animal and human, they usually create a carry-on type of the lesion. Carry-on means, ma'am, it is like means severe lesion of the ringworm where we can see even the blistering, and it is very common now in the humans. Very common.
Okay.
Few of the human cases in the PGI Chandigarh. Earlier, they were suffering from the ringworm infection in the groin area. Later on, it spread to the entire thigh, then entire stomach, then entire body, and now they are under the in the ICU, and they are going for the intensive therapy because of the secondary bacterial complication. This because of the ringworm along with the secondary bacterial infection. Now the clinical scenario of the ringworm or dermatophytosis in the human is entirely changing.
Okay.
So
Thank you.
main agent is the this trichophyton mentagrophyte in the humans, which is causing havoc. But Microsporum gypsum, when it is causing infection, there are few reports. There are few reports that this is causing infection in the human. Then they are producing a severe lesion, more severely.
Okay. Thank you. So now the next question is, what is the link of cryptosporidium with diabetes mellitus as was shown in a paper?
Cryptosporidium.
Cryptosporidium. Yes.
No, ma'am. I think it is Cryptococcus.
Okay. Okay.
Yeah, Cryptosporidium is parasite. This is a
Yeah.
Cryptococcus.
Yeah. It is a protozoan parasite. Yes. Yes.
Yes.
Okay.
Somebody
Then,
has asked.
yes.
Just a minute. I will open the chat box.
Yeah.
Okay.
Please. Dr. Safida.
Okay, a lot of questions are, what is the link of this with diabetes mellitus as shown in the paper? What is the link of Cryptococcus? This Cryptococcus neoformans is the like disease which mainly affects the immunocompromised host. If we talk about the human, so the diabetic patient may have a more chances to get the infection with the Cryptosporidium. Okay, sorry, this Cryptococcus. Then Mona Priya is how for blastomycosis in dog incidents. Blastomycosis in dog, there are few reports from the India, but still they are not confirmatory report. But blastomycosis in humans, they have a lot of reports from India, especially in the PGI Chandigarh, as well as from the PGI Raipur. And then any confirmatory diagnostic test for the blastomycosis, we do not have any confirmatory diagnostic test. We need to go for the combination of the culture as well as the any advanced technique like VTEC system or any this blastomyces usually is a normally we diagnose it on the pattern of tuberculosis. Like blastomycin, we used to produce the like tuberculosis, we used to produce the blastomycin. So in blastomycin, we'll inject in the entire flock or in animals, and then we will suspect whether this agent present or not. So there is a no test for the confirmatory diagnosis. Okay, then Dr. Safida Begum, sorry, the black fungus and white fungus which came into importance soon after corona outbreak, zoonotic. Yes, definitely they are zoonotic. Black fungus is nothing is the mucorales. White fungus is nothing. It is a candida. So there is yellow fungus also, which also belongs to the mucorales. So black fungus is 100% zoonotic. They are the main class of the mucorales. Though Cunningham mela has been reported is causing highest mortality, not in India, but it is reported from the abroad countries. This black fungus like mucor, rhizophus, there are many other. So they are main agent in the black, we can say the black disease during the in the COVID patients, where the blackening of the tissue used to occur, and mainly the this mucor and rhizophus. White fungus is a candida infection. Is same. It is causing the oral lesion as well as the otitis is the white fungus. Then what is their origin of infection in human? It is nothing like it is a because of the use of contaminated syringes and all that. So these are the always present in the like in the soil or as a saprophyte. So they during the immunosuppression they usually cause the disease.
Next question is by Dr. Ankita. She is
Yeah,
asking, sir, do you think the fungal infection can be followed by bacterial diseases?
yeah.
As
Why
once we treat bacterial infection and it may show fungal.
Sir, do you think the fungal infection can be followed by bacterial disease as once we treat bacterial infection and it may show fungal? Mm. I am not getting what you are asking, Dr. Ankita or Ankita, who is me.
Do you think the
Do you think the fungal infection can be followed by bacterial diseases? Why not? Any bacterial disease can be followed by the fungal disease and vice versa. And once we treat bacterial infection and it may show fungal. See, when you are using antibiotic to treat the bacteria, there is a possibility that fungus may survive, fungus may spread, and it may cause the disease. And if you are suffering from the fungal disease originally, there is also possibility that you can get contaminated with the bacterial disease or cross-infected with the bacterial disease. And it is a natural phenomena going on in the animals as well as in the human.
Yes. So, Dr. Abhishek, I think there are no more questions. Thank you so much for answering all the questions with a lot of patience, and it was good discussion.
Thank you very
So,
much, ma'am.
yes, if there are no more questions, any other participant? I don't think so. Then I think we can close this session. And in the end, I would once again, thank you for agreeing to deliver the lecture on a very important topic, that is mycotic diseases. And
Thank you very much, ma'am. Means my sincere thanks to ICR Ivory Institute Palampur as well as the NADCL for giving me this opportunity to interact with the participant, especially on the mycotic diseases, which is like very neglected, especially in the animal side. Thank you very much, ma'am.
yes, thank you, Dr. Abhishek.
Thanks to one and all. Thank you, thank you. Ma'am, may I leave?
Yes, Dr. Abhishek.
Yeah. Thank you very much, ma'am.
Yeah,
Thank you.
thank you. Thank you. To the participants, I would like to say that you will be receiving the assignment one by today, and you'll have to submit it maybe by tomorrow evening or Monday. So for tomorrow, you'll be receiving an assignment. Thank you, one and all. Thank you.